Welcome everyone to the first 2300 review video. This video will be an overview and review of antigens and antibodies, and we'll talk about the basics and terminology first, and then work our way into the significance and various roles that antibodies can play in the immune system. And we'll also take a look at the different characteristics of the 5 major classes of antibodies that you'll need to know for your next exam. So you want to make sure that you know these basic terms and definitions, like what are antigens, what are antibodies, B cell receptors, T cell receptors, and antigens can pretty much be anything that reacts specifically with an antibody or a B cell receptor or a T cell receptor. And we have self antigens as well as are being constantly exposed to foreign antigens. And our immune cells are going to recognize antigens as foreign when the epitopes of that antigen bind to epitope-specific receptor regions on, or these molecules on B cells and or T cells. Whereas our self-antigens are typically not going to bind with our body's immune components, except during the case of autoimmune disorders, which we'll talk about in the next video. So, antigenic determinant is the term we use for the specific site on an antigen that is recognized by components of the immune system. So, when we say antigen, each pathogen has hundreds to thousands of these antigens. And if you zoom in, each of these antigens have 1 to dozens of epitopes, which are also referred to as antigenic determinants. These two terms are interchangeable. And these epitopes are these little 3D regions of antigens whose specific shapes are recognized by immune receptors. So it's sort of like the key to your lock. And these antigens can be categorized based on their relationship to the host cells. So exogenous antigens will originate from microbes outside of our body cells, so exo, outside, whereas endogenous antigens are going to be produced by intracellular microbes. So we're talking viruses, we're talking OIPs, obligate intracellular parasites, like Chlamydia trachomatis or other intracellular pathogenic bacteria. And we are going to find these endogenous antigens incorporated into a host cell's cytoplasmic membrane, but we'll take a closer look at that in another diagram coming up soon. And we also have autoantigens, which are our normal cell antigens. So going back to antigens, so we talked about what an antigenic determinant was. An immunogen is specifically an antigen that's going to induce an immune response. So it has to be able to mount a response, which not all antigens are capable of doing. And a great example of that, and we're going to see this with certain hypersensitivities, and we'll see it again with immunizations. But oh, what just happened? Okay, so we're not necessarily going to see a mounted immune response with all antigens. So we'll see this happen with haptens, and basically haptens are just molecules that are too small to mount an immune response on their own. So typically they're going to be coupled or conjugated with a carrier molecule like a protein. And proteins and big polysaccharides induce a pretty strong immune response. And so conjugating a very small hapten, even though it might be antigenic in that an antibody might be able to recognize it, it still might not be immunogenic. And the way to make a hapten molecule immunogenic would be to couple it with something that is going to induce a strong immune response. And lipids and nucleic acids usually are not as immunogenic. Um, so just keep that in mind. T-dependent antigens versus T-independent antigens. So a lot of the antigens that we're talking about today, when we say antigens, these are going to be the T-dependent antigens, and they're going to be the ones that B cells are going to require the assistance of T helper cells to be able to be activated. And this is the mechanism that's evoked by proteins and haptens bound to proteins. The response is going to be pretty strong. It's going to be long lasting, and it's going to involve your memory cells. And it can be boosted. Whereas T independent antigens are not going to involve the help of T helper cells, and it's usually going to be large molecules with lots of repeating units like polysaccharides, and they can't usually bind to MHC molecules and be presented on them. So you might think, okay, well, that just sounds like you're cutting out a middleman. Wouldn't that make for a more efficient response? But actually, the antibody that's usually generated from T-independent antigens generally have a lower affinity for its antigen, and so it's usually in circulation for a short amount of time. And it also doesn't produce memory cells, so they're going to mature more slowly, and that's also going to cause you to be a little bit more susceptible to bacterial infections earlier in life. And it's kind of why we'll see that again with why we can't use purified polysaccharide vaccines for kids who are too young, like younger than 2 years old. They're just not as effective, so we might have to use conjugated vaccines with them. But we'll talk a little bit more about that when we get to immunizations. Okay, so like we said, antigens can not only bind to antibodies, which we'll take a look at in just a second, but they can also bind to B cell receptors or T cell receptors. So B cell receptors, which we see on the right over here, can bind directly to epitopes on proteins, polysaccharides, nucleic acids. Even though they're way more likely to bind to antigens on proteins and polysaccharides. T cell receptors on the left, like we see over here, they're going to be embedded in the membranes of T4 lymphocytes and T8 lymphocytes, which are only going to be able to recognize epitopes that are presented by our body's molecules on these MHC presenting platforms. So just want to point out when you compare the structure of antibodies to these receptors, they're quite similar. So you're going to have a really specific epitope binding site on these variable regions. You also have variable regions here on your B cell receptor, and that's really going to be the primary difference in these B cell receptors versus free floating antibodies is the B cell receptors are actually going to have this transmembrane portion. Whereas when we say free floating antibodies, they're not actually embedded on a cell, on the surface of a cell. And T cell receptors they're quite different in structure to begin with. Okay, so what are antibodies? So you might see some sources sort of use them interchangeably, and it's okay to use them interchangeably for now. But if you want to get real technical with it, technically, immunoglobulin is the term that we use for the protein molecule that has that 4-chain structure with the 2 identical light chains and the 2 identical heavy chains. And we use antibodies for the term for the soluble protein that's not bound to a cell surface. And these chains that we're talking about, it's going to have a Y shape, and these chains are going to be held together by disulfide bonds to form an antibody monomer, mono meaning one. But as we'll see, some of these can take those monomers and form can take multiple of these monomers and create more complex structures with them. So we're going to take a look at the 5 different antibody classes. And when we say classes, what we're technically saying is we're looking at the different isotypes. And the isotype or the class is going to be determined by the constant region of the heavy chain. So each antibody class is going to have a different constant region of the heavy chain from another class, but all the classes, all the antibodies within the same class will have the same constant region of the heavy chain. And so this region is basically the Fc region. It's going to contain the Fc region. It's the portion of the antibody that determines the biological function, which is basically going to be its ability to bind to receptors on phagocytes, on mast cells, on basophils, and its ability to activate the complement pathway. And like we said, it's going to reside in that Fc portion of the antibody. All isotypes can exist as monomers. That's like the simplest sort of unit, but like we said, we're going to look at some structures that they can form with multiple monomers. So we're going to look at the Fc fragment that's of the antibody that's going to be indicative of the function of that specific antibody class, and the FAB region is going to help determine the specificity of that particular antibody, because the FAB region which we see here is going to include this little portion right here that I'm pointing to with my mouse. And that is going to be our variable region. And the variable region is going to be the portion that contains the site that's going to bind to the antigens we just talked about. And most antibodies are going to have two antigen binding sites on the arms of that Y structure. And each plasma cell has the potential to produce different classes of antibodies, but they can also undergo something called a class switch, which we'll see in just a second. But typically, mature naive B cells express IgM first, and IgD. Sometimes they show up simultaneously on the surface of these B cells prior to activation, but then once they go to the lymph node and once they have exposure to the antigen and interact with other immune cells to undergo the isotype switch, they can switch to IgA, IgG, or IgE, and that, like we'll see in just a second. Okay, so I thought this was a cool picture of a close-up of an antibody molecule. An antibody molecule is, so this is a protein in itself. And it's made up of various glycoprotein chains. Like we said, 2 heavy chains, 2 light chains. The FC portion right here is the part that's going to be responsible for the biological activity of the antibody. And this portion is pretty much going to become biologically active after the FAB portion, the FAB portion right here has bound to its matching antigen. Depending on the class and subclass of the antibody, biological activities of this FC region can range from activating the complement pathways to binding to receptors on phagocytes or mast cells or NK cells, and they can even determine the tissue distribution of the antibodies, like sort of be a guide for telling them where to go. And this is just a slide of a drawing that I made on Paint to color code different antibody molecules to show you the breakdown of exactly what we mean when we use these terms. Just kind of simplified here, because when you see it all at once on one antibody molecule, it can get a little confusing, and I know a lot of you were sort of getting a little bit confused about the whole FC and constant versus variable region thing. So structurally, the purple portions are the heavy chains, and this heavy chain is identical to this heavy chain, and this light chain is identical to this light chain. Again, joined together by disulfide bonds. So the pink here is all of this pink here is going to represent our FAB region, our antigen binding region, and all of everything from the hinge to the entire lower half of the antibody molecule will be our FC region. And within our FAB region, we're going to have this orange portion, which is going to be our variable region, but a portion, the lower half of the FAB region is actually going to be grouped with the FC region under the constant region. So when we say constant region, we should specify whether we're talking about the FAB region of the constant region or the FC region of the constant region. So this is just kind of showing you where on the antibody molecule we're actually binding. So this is a close-up of a picture that kind of shows this cross-linking. And I just want to make sure that you know where specifically on these molecules we're binding and why it matters, because it's incredibly specific. So when we say binding of the antigen, we're specifically talking about the arms of this Y-shaped molecule in a very specific place of the variable portion of the heavy chain and light chain. Whereas when we talk about signaling or binding to receptors on cells of our immune system, we're talking about this specific portion, the FC portion, that's actually binding to those FC receptors of these phagocytes. And this is just another close-up here. So FAB binding to these epitopes, the FC once, and it has to be the FAB portion of these antibodies have to bind to the epitopes of this microbial antigen before it can actually use its FC portion to attach to the FC receptor that's on the surface of the phagocyte. And it needs that attachment to the antigen for enhanced attachment. And this is especially important against microbes that have a capsule, encapsulated microbes. So what exactly happens when antibodies and antigens bind to each other? So we're going to look at 6 different possible outcomes. So we can have neutralization occur, and someone asked me, what is the difference between neutralization and something like agglutination or precipitation? Because from the outside, doesn't it look like you're just taking those antibodies and binding them to these antigens? Well, specifically agglutination and precipitation, we're talking about cross-linking, and that could be cross-linking to multiple pathogenic cells or multiple even to each other, so multiple antibodies. But that sort of clumping that you'll see occur, whereas neutralization would be more like disarming the weapon, if you will. And so these weapons that we're neutralizing can be toxins, they can be viruses, they can be surface antigens on a bacterial cell. But basically, if you coat them with an antibody, they have no means of binding to the surface receptors on our cells. And as we saw with these pathogenesis of viruses back in the basic virology unit, they have to be able to get in first. We have to let them in and they have to enter the cell to be able to actually start to replicate in the case of a virus. So if we block that, then they can attach and they can't establish that intracellular infection and go on to replicate. They can also bind to specific portions on cell structures. So, and when they do that, it's actually going to interfere with whatever the function of that cell structure is. So, for example, if the antibody binds to an antigen on the flagella, it can interfere with motility or the movement of that cell, or if the antibody binds to, say, pili. Remember, pili are in Fimbriae are used for attachment, which is the very first step that a microbe needs to be able to do to establish an infection. So you're basically kind of nipping it in the bud before it even gets a chance. And agglutination and precipitation is pretty much when you're going to see both arms of that antibody binding multiple antigens. So they can be creating these complexes with soluble antigens, or they can sometimes cross-link with each other. And we'll take a look at a little bit of that when we talk about that cross-reactivity. Or they can bind to separate but identical antigens on the same cell or on different cells. And basically, it's just going to create this giant clumpy mess. And it pretty much flags our backup to come and more easily phagocytize it. So we talked a little bit about opsonization before in previous lectures. Dr. Byrne referred to these opsonins as sort of like sprinkling salt on your food. Why do you sprinkle that salt on it? Or I guess in my case, it would be Tabasco sauce because I put that on everything. But we put it on our food to make it more delectable to eat, right? So basically, you can think about it as sprinkling this bacteria with salt to really make it yummier for that giant phagocyte to come along and swallow it up. And there's specifically one antibody class that we're going to talk about with opsonization, but a bit of a throwback. Who can remember the specific complement protein that was like the number one complement protein that we had to know that was going to be like the primary opsonization complement protein? I'll give you a second to think about that. And that's going to be our C3b complement protein. So antigen antibody binding can also activate complement pathways. So in the case of IgM and IgG, that can activate our classical pathway. And for some of the others, we can also activate the alternative pathway or the lectin pathway. And if you think back to what the effect of what the three possible effects of uh, complement were back in the innate unit, that was that the cell could lyse or burst. You can also induce inflammation, which basically is just going to involve our cells generating inflammatory mediators like that IL-1, IL-6, TNF alpha, and opsonization with that C3b. And we can also induce ADCC. So that's going to involve our NK cells interacting with our IgG immunoglobulins, or antibodies, sorry, and induce antibody-dependent cellular cytotoxicity. So it's what happens when the NK cell makes contact with the IgG, and they start to release these perforins and granzymes, which are just proteolytic enzymes, and these granzymes are going to pass through the pores and activate apoptosis, which is programmed cell death, which is pretty much like going in and hitting that self-destruct button. And as a result, it's going to pretty much break this down and just become pieces of debris, and then our phagocytes will come and clean it up. So this is just a quick review of the 3 different complement pathways and the effects that they can have. Quick side note, want you to remember that, the central complement protein to all 3 of these pathways is going to be our C3b molecule. Very, very important. The only one that is specific to this interaction with antibody is going to be the classical pathway. So, that's going to take a little bit of time for our cells to be able to activate our adaptive immune system. So, basically our lectin pathway and our alternative pathway, there are different means of buying this one a little bit more time. And C3A here is, along with C4A and C5A, are going to be great inflammatory complement proteins called anaphylatoxins, which we'll talk about in the next video. This is a table out of Bauman, your textbook, sort of comparing the different antibody classes. So let's get into IgM. So IgM is super special because it can actually take that monomer, which you might find on the surface of a B cell, and it's pretty much the only type of Ig that you'll find on any surface of an immune cell with another antibody class. So sometimes you'll see IgM with IgD concurrently on the surface of that B cell, but those are the only ones that you'll find on the same surface together. So you can see it as a monomer on those surfaces, and they're going to act like sort of B cell receptors, but they can also be joined together to form this pentameric structure. And just want to point out, notice how they're all joined by the constant region. And you want to do it that way because you want that FAB region pointing outward to be able to bind to antigens. And this is really great because uh, first, it's the first class that's made during the primary immune response, so it's really nice to be able to have something so um, effective at binding, because it's going to be that first class and really the principal class also that's produced during T independent antigen response. But it's the first one in response to the primary immune response. And so um, a lot of times we can measure the levels of IgM um, early earlier on than the other IGs that serve similar functions as IgM. Um, and so it can be indicative of a more recent infection. Um, and IgM is, there's only 5 to 13% of it in circulation. And it's usually found mainly in the bloodstream rather than in the tissues, because it's usually too big to cross into the tissue. So if you look at the relative sizes, and you're not going to need to know or memorize these exact size numbers or anything like that, but just wanted to show you as a point of comparison how big these are compared to the other antibody molecules, because I want to come back to that when we talk about IgG. And because it forms this pentamer, you have 10 possible epitope binding sites, which makes it a super efficient cross-linker and complement pathway activator. And the whoopsie, sorry about that. My mouse went a little crazy there. But okay, so as I was saying, so this IgM can form a pentamer. So it's got these 5 monomers into one complex, and that makes it super efficient at what it does. And it can neutralize, it can cross-link, it can, and it's the first one made, so it's the one that's going to be able to class switch into IgA, IgG, and IgE. And the way that you can think about that is the acronym AGE. So IgA, IgG, IgE. So IgM first, and then it'll class switch into those 3. But because it can cross-link, it's also that means potential for clogging. So it's a major component of rheumatoid factors, which are basically autoantibodies, which are basically antibodies that are going to react to our own antigens. And so sometimes they'll end up clumping together and cross-linking into this complex with other antibodies. And that's going to cause a really big issue in places like synovial fluid in joint spaces and things like that. And we will take a look at a video of that in just a second. This is also the only antibody that can be formed by the fetus in utero. Now, normally the fetus doesn't make any antibodies when it's in utero. Normally, it pretty much lives with the passive immunity that it receives from the mom. So the mom can pass on IgG antibodies to the fetus, and those maternal, because they're so long lasting, those maternal antibodies will protect the baby after birth, even for a while, for months after the baby's born. But then after that, IgM will be the first antibody class that the baby will start to make. But if there's some sort of pathogen that invades that can cross the placenta and try to infect the fetus, IgM technically can start to be formed upon exposure to an antigen like that. It can also activate the classical complement pathway, and I think that's all I wanted to say. So, let's watch that pathology in action.
Rheumatoid arthritis is a common autoimmune disorder of the joints that affects about 1% of the population worldwide, more often women than men. The exact causes of the disease are unknown, but an individual's risk is thought to be increased by a combination of genetic and environmental factors. Many of the genes associated with rheumatoid arthritis are involved in immune system function. Non-genetic risk factors include age, diet, infectious agents, and smoking. In an autoimmune disorder such as rheumatoid arthritis, the immune system mistakenly attacks and destroys the body's own cells and tissues, which immunologists refer to as self. And this autoimmunity can begin many years before joint symptoms are noticed. Doctors can use blood tests to identify this preclinical phase by the presence of multiple factors in the blood, such as autoantibodies and inflammatory factors. These early autoantibodies are thought to first develop outside of the joints, possibly in the gut, mouth, or lungs. Environmental factors such as smoking can modify our self proteins, making them targets for the immune system. One particular modification is called citrullination. When the immune system recognizes these modified self proteins, it leads to a breach of self-tolerance and the production of autoreactive B cells and autoantibodies, and later autoreactive T cells, the hallmarks of autoimmune disease. By the time that symptoms appear, the immune response has intensified and the antibodies produced target a broader range of self-proteins. But it's only in some patients that this systemic autoimmunity progresses to joint inflammation, which is possibly triggered by an increase in joint permeability and increased access to antibodies. Working out how to prevent this progression is a major goal for scientists. In patients who do develop joint disease, the small joints of the hands and feet are most commonly affected. After entering a joint, immune cells and autoantibodies bind to modified self-proteins in the cartilage, bone, and lining of the joint called the synovium. This induces an inflammatory response and activates cells in the joint such as macrophages, neutrophils, and osteoclasts, as well as blood monocytes. Activated monocytes differentiate into yet more macrophages, which together with other cells in the joint produce soluble inflammatory factors known as cytokines. Drugs targeting these cytokines are highly effective treatments for many patients. Left unchecked, however, damage caused by the inflammatory environment can expose new self-antigens to the immune system, continuing the cycle. At this stage, the first clinical symptoms of joint pain, swelling, and warmth appear. Some patients recover, but most commonly, patients develop a chronic destructive disorder. As the disease progresses, dendritic cells display the newly exposed self-antigens and activate T cells in the joint itself or in the local lymph node. In addition, B cells infiltrate the joint, where they proliferate and produce antibodies and other factors, further amplifying the autoimmune response. Cells in the joint lining, called fibroblast-like synoviocytes, also proliferate and grow into the joint space, spreading across to the cartilage surface. These cells secrete matrix-degrading enzymes which erode the cartilage tissue. Bone is also eroded as osteoclasts, which contribute to normal bone turnover, become hyperactivated. Bone erosion, cartilage destruction, and joint swelling cause severe pain, restrict movement of the joint, and in the worst cases can cause joint deformities. And as well as the joints, other organs and body systems can be affected by the ongoing inflammation. For example, inflammation in the blood vessels can lead to heart disease. So early and aggressive therapy is recommended to prevent these systemic complications. By further understanding the immune markers and mechanisms of rheumatoid arthritis, it's hoped that targeted interventions could ultimately change the course of the disease.
Okay, so now let's get to the IgG class. And this is pretty much going to be a monomer, but it's going to be one of the most important classes. It's going to be what makes up a majority of the antibodies in your serum. And eventually it's going to circulate, but it will exit the blood vessels to enter tissues. And it has the longest half-life of all the classes. It's the primary one during the secondary response, which is why that makes it the antibody of memory. It's also the only one that can cross the placenta, and so it's what's going to confer that passive immunity to the from the mother to the fetus while in utero. And it can also pretty much, it's sort of the jack of all trades. So it can do that neutralization that some of our other antibodies like IgM and IgA can do. It can also activate the classical complement pathway like IgM can. But it can also opsonize and as also bind to NK cells to induce ADCC. And it is the only one that can cross the placenta. The other classes can't do that either. So let's see. So the FAB portion up here of this IgG is going to bind to the epitopes of an antigen, and the FC portion right here is what's going to bind to the phagocytes. And act as opsonins, just like C3b. And that's particularly important for encapsulated bacteria. So if you think back to sort of a side note, but also a quick recap, our 3 bacteria that Dr. Byrne talked about before that had the juiciest capsules included bugs like Streptococcus pneumoniae, our extra diva Haemophilus influenza, which does not cause the flu. It is a bacterium that is extremely fastidious. And last but not least, our Neisseria meningitidis. So you can also find IgG in breast milk, the very first breast milk made after birth, just like you can with IgA. So the big takeaways, it's pretty much when you see an antibody class referring to the memory response or a secondary immune response or something about crossing the placenta or opsonizing or inducing ADCC. That's pretty much going to be indicative of IgG. If it's talking about breast milk, it could be either IgG or IgA. And IgG is also the most significant class as far as neutralizing goes, because for neutralizing during the secondary immune response goes, because these IgG responses are often going to result in lifelong immunity against a lot of these pathogens during the secondary response. Next, we have our IgA, and the special thing about that, that I also forgot to mention, is want to point out this is one of two antibody classes that has a J chain. So pretty much if you're asked about an antibody class, an antibody molecule that will have a J chain, that's probably either going to be your IgA or your IgM. So your IgM also has a J chain, but it's the only one that's going to have a secretory component. So one thing you absolutely want to make sure that you know is that it's got this S chain. Like the others, it can be a monomer when it's in the serum, but a lot of times you'll find IgA more in the secretions along the linings of mucous membranes. And our mucous membranes are pretty much going to be found in our gastrointestinal tract, our genitourinary tract, and our respiratory tract. So you'll find it in saliva, in mucus, in tears, in your mucociliary escalator, your breast milk. It's going to have this molecule called secretory IgA, which means it's going to have that S piece. Sorry, that should say piece. And basically, the IgA will be present in these mucus secretions because it'll protect these internal body surfaces that are basically exposed to your external environment. By blocking the ability of these bacteria and viruses to attach to your mucous membranes. So that means it's capable of neutralization, and it'll confer mucosal immunity by blocking them. And it's also this FC portion of the IgA will actually help bind to parts of the mucus, and so it can actually help trap microbes. And the secretory component also helps protect it from digestive enzymes in the secretions. And any reaction of IgA with an antigen in the mucous membrane can actually initiate an inflammatory response that will help mobilize and call for backup and mobilize other immunoglobulin and cell defenses to that site of invasion. And the FC portion of the secretory IgA can also bind to macrophages and neutrophils for enhanced attachment, and the IgA can also activate the lectin complement pathway and the alternative complement pathway. It doesn't have an FC domain for activating the classical complement pathway, so only IgG and IgM are capable of that. Oh, and also it's produced by plasma cells that are present in your MALT, which stands for mucosa-associated lymphoid tissue, which is going to be pretty much these concentrations of lymph tissue where you'll find a lot of your lymphocytes and your bodyguards that are pretty much there to help defend your body. And you'll find mucosa-associated lymphoid tissue like toward the end of your small intestine, so like lining in the mucosa of your ileum, places like that. Okay. So next is IgD, which is pretty much the class that we know the least about, but it's for the most part gonna be a monomer, and you're pretty much gonna see it primarily bound on the surface of B cells, where it's pretty much gonna function as a receptor for antigens, and they think it might help control B lymphocyte activation and suppression, but it certainly is responsible for the development and maturation of antibodies, and it may play a role in eliminating B cells that are generating self-reactive autoantibodies, so might have its hand in all that, but it's only going to make 0.2% of your serum antibodies, so there's not going to be a whole lot of it in your serum. Like IgE, there's very, very little of it circulating. IgE is also found as a monomer. It makes up about 0.002% of your serum antibodies, and it only has a half-life of 2 days. So both IgD and E are pretty short half-lives, and there's very little of it normally circulating in your serum. But these are particularly special because they're FC regions. So basically, the antigens to things like helminths, which are parasitic worms, and arthropods, and even allergens, will bind to the FAB portion of the IgE. And when it cross-links, it triggers the release of vasodilators from cells such as basophils, mast cells, and eosinophils. And so those guys are capable of secreting things like histamine, cytokines, heparin, basically things that are going to induce vasodilation and bring more blood and whatnot to the area to call for backup. So you're going to see inflammation. It's also going to help enable your IgG and complement proteins and leukocytes to come to the tissues and trigger coughing, sneezing, things that are going to help you mechanically remove those microbes and toxins. And it's also going to be responsible for your anaphylactic hypersensitivity. So let's take a look at that in action really quick.
Some people develop an allergic reaction or hypersensitivity when exposed to substances such as dust, pollens, animal dander, or penicillin. This hypersensitivity is mediated by IgE. Sensitization occurs when the antigen makes contact with some part of the body. The antigen is taken up, processed by antigen-presenting cells, and presented on a class II MHC to T helper cells. Tissues under the mucous membranes are rich in B cells committed to IgE production, and IgE-producing cells are more abundant in persons susceptible to allergies. The T helper cells produce cytokines, which stimulate these B cells to proliferate and differentiate into IgE-producing plasma cells. As IgE is produced in specific areas of the body, the IgE molecules attach via their constant regions to receptors on nearby mast cells. Mast cells contain granules packed with chemicals that induce a hypersensitivity response. Once attached, the IgE molecules can survive for many weeks. The individual is now sensitized to the antigen. When exposed to the antigen for a second time, the antigen binds to the IgE antibodies on the mast cells. To trigger a response, two cell-bound IgE molecules must react with a specific antigen. Within seconds of the reaction, the mast cell releases histamine and other mediators of the inflammatory response from the granules, triggering a variety of symptoms.
And now that we've talked a little bit about the different antibody classes, let's take a brief look at how these antibodies are made. So just a quick recap, you need to have that antigen binding to the APC, our antigen presenting cell, which remember we said includes our 3 MVPs, B cells, which we see here. My personal favorite, dendritic cells, DCs, and macrophages. And these APCs are specifically when we're looking at the multiplication of antibodies, you'll have a B cell present that antigen on an MHC2 molecule and present it to this T cell, which will then go on to serve its effector functions, such as activating phagocytes to have enhanced phagocytosis, or to create plasma cells and activated B cells that will make plasma cells, or memory B cells that will be very long lasting, relatively, or these plasma cells that can crank out tons and tons of antibodies very fast. So, quick recap. Remember, the function of these T-dependent antigens were to enhance the activation of these B cells to they can serve that purpose also when it needs a second opinion. If it is exposed to a T-independent antigen, then it may also still go to a T helper cell to wait for cytokines released from the T cell that will tell it to go on to activate. But basically, just know that it leads to B cell activation. That's what you have to have before you can start cranking out all those antibodies. Okay. And there's really just two things that I want you to make sure that you take away from this, because this is just a graph of the primary immune response and a secondary immune response, and basically what we should expect to see as far as antibodies go in our serum. And so if we, for example, were to look at the tetanus toxoid, which we know toxoids to be pretty much inactivated exotoxins, which can pretty much be used as a vaccine so that it can induce an immune response. And so you will see a bit of a lag period for the first couple of days, because you're not going to see any antibody in the serum then. It's going to take some time before you even see IgM, which will be the first antibody on the scene, but there's still going to be a lag period for the very first primary response. And once you've gone through the recognition of the antigen and the processing of the antigen and then the specific activation of those immune cells, it's then going to, any macrophages will present those antigens to the T cells, and it'll cause B cells to be activated, and then the plasma cells that result from that are going to start producing more IgG and a peak of IgM. And notice that the peak for the IgG is, it takes a good about 2 weeks or so before IgG to really kick in. So that's for the primary response. But then if you compare that to the secondary response, not only is there not a lag period, but IgG responds pretty immediately. So the two things I want you to take away, that it's a much faster response the second time around when it's re-exposed to that antigen again. and also the fact that you have a much greater magnitude in the response. Look at how high this is compared to even the maximum of this. And that's a result of the secondary response involving a greater proportion of IgG antibodies being cranked out. So basically, the antibody forming system is like a learning system that responds to each exposure by producing large amounts of a specific antibody. And I thought this was a really cool graph that sort of shows you what's going on with these lymphocytes side by side to the antibody concentration graphs across time. So after the first exposure, you're going to get what we call a anamnestic response, which is like pretty much your booster response, right? You're going to see a faster and you're going to see a stronger response from your adaptive immune system after a first exposure. So, also, I really like this diagram because you can see how, you're pretty much starting out with IgM in these B cells, but then at some point you are going to experience that class switch to the IgG. But like we said, that's going to take some time. And you can also see your memory cells taking part during the primary response. And towards the sort of end of that primary response. And then in the secondary response, you see a much greater proliferation of your IgG. So we've pretty much touched on that. And one thing that I do want to kind of tie this back to is bringing it back to the beginning when we talked about pathogenesis and virulence factors of these bugs and whatnot. One of the major virulence factors of a major bug that we've studied extensively actually has found a way to work around this. Which is pretty ingenious. So some of these microbes have found a way to circumvent our host immune system through the cranking out of Fc receptors, just like our immune cells do. And so Staphylococcus aureus can do this, some streptococci, HSV, herpes simplex, VZV, CMV, but basically by producing these Fc receptors, they can bind IgG backwards. And so this is one of the primary virulence factors of Staphylococcus aureus. It has that protein A, which is pretty much going to act like that Fc receptor that our cells have. So it's basically copying our immune cells. And what it's going to do is it's going to bind the wrong end of it. Because what it's supposed to do, which you can see on the left here with Staph epi, it's supposed to bind this recognizable antigen on that FAB portion. And it's the FC portion here that's supposed to bind here. But now that Staph aureus has this protein A molecule, it sort of wears it upside down, and our phagocytes are not able to recognize it. So it just moves on, totally camouflaged, able to continue on to cause an infection and go on to continue. And so basically, and another thing is because it's binding the FC portion of the IgG molecule, it's also preventing the activation of complement. And so you're not going to have C3b produced, and you're not going to have that opsonization and phagocytosis. And it's just a really, really smart way to get around that. So this is one of the primary valence factors of Staph aureus. And so because none of the other coagulase negative staph have this as a virulence factor, sometimes clinical laboratories will actually use this in certain tests because it will bind to IgG and form an antibody complex with it, like you see here. And that's pretty much it. So you can, so I've pretty much I was planning on making this in an SI worksheet, but I just pretty much put that whole activity on this slide. So you can try this out and feel free to email me your answers. I did go over this in SI last week, but for those of you who maybe couldn't attend that SI session, please feel free to try this and send me your answers, and I'm more than happy to give you feedback on them. For some of these, they're not all only going to refer to one. You may have more than one antibody class that applies to some of these. So, for example, we learned earlier that, for example, 20 can activate the classical complement pathway. So that's not only going to be IgM, which is really efficient at doing that, but it's also going to include your IgG as well. So watch out for that. A lot of this is in your Bauman textbook. You can also, Mile, again, is a great resource. You might find some of these on Dr. Burns' slides on the adaptive immune system, but I definitely just make sure that you practice applying. Because I know that, especially for those of you who have taken AMP2, this might seem very familiar to you, but just try to continue to practice connecting these concepts to the other chapters about virulence and pathogenesis and host evasion and immunodeficiencies and hypersensitivities.